Thank you very much. So I have plenty of slides, so I will rush through my old drugs. I can tell they're terrible. So that's my disclaimer, and I will be addressing the priority critical pathogens, Acinetobacter baumannii, Pseudomonas aeruginosa, that is carbapenem resistant, and Enterobacteriaceae that are either CRE or ESBL producers. I'm sorry. So colistin is used over the last decade, as 2 decades, as the go-to drug agent to treat carbapenem-resistant gram-negative. Correct dosing was figured out maybe in the last decade only, and accurate susceptibility testing is still not figured out. It's an unresolved issue. Most laboratories do not provide accurate results for colistin susceptibility. Its efficacy is doubtful, and it has serious side effects. And I just brought few examples. That's, again, the wrong dosing we know now, but that's a study, I think it's from South Africa, comparing colistin to tobramycin to treat pneumonias. We all use tobramycin as our usual drug to treat pneumonias, of course. So it's things that we would not do on a regular basis, of course. But surprisingly, it was better than colistin, and the study was stopped because better results with tobramycin compared to colistin. And then another study of amsulbactam versus polymyxin, and here you see the death rate are extremely high in both groups, but are actually worse in the polymyxin group. And this is looking again at the same things here. It's various comparators to colistin, and you can see again greater mortality with colistin than with other agents. Not only that, but colistin resistance is on the rise, and that has been seen specifically in carbapenem-resistant strains, and it became an epidemic both in Italy and Greece, and maybe in other parts of the world that we don't know about. And so I would say that over the last decade or 5 or 6 years, people have opted to move to combination therapy, and the rationale for combination therapy is that colistin has unproven efficacy, there is heteroresistance to colistin, time to reach appropriate level with colistin are long, and various agents have been recommended, but I would say that the most common combination that is used is actually a combination with carbapenem. In vitro and in vivo results are actually non-consistent between themselves, and it may reflect strain-to-strain variation, and human data is limited. So, and here also a little bit of data. There are multiple retrospective studies that shows that combination therapy is better than single colistin alone. And here you see one of the studies showing monotherapy you have 54% mortality, with combination therapy 34% mortality. The problem with all of those studies that they're retrospective, and when we did look at the results of those studies, you can see that when you look at each combination compared to monotherapy, you see no difference. So colistin, carbapenem, colistin tigacycline has exactly similar results as colistin alone. Only when you look at the colistin versus mixed comparator, you see the effect of better activity of or better results with combination therapy, and that suggests that that's actually a bias in the studies rather than the true effect. There are 2 large studies that are conducted now. One has just finished. In this study, it's a study that was funded by the European Commission comparing colistin to colistin meropenem, and we included about 400 patients with, and I would say about 300 of them were Acinetobacter, which is carbapenem resistant, and a few Pseudomonas, and about 80 or 85 CREs. And the patients were extremely sick. They were, really real-world data with very high mortality rate. And you can see that even within 48 hours, there was 6 to 7% mortality rate, so very sick patients. And, and what you can see here, that there was truly no difference in mortality between the two arms, and quite high mortality, 28%. At 28 days, it was close to 40% mortality rate. So no benefit to combination therapy. Of course, that will not change the practice, as I've mentioned to David before. There is a very similar study that is done, funded by the NIH and conducted by Keith Kay, and will take probably another year and a year and a half to get the results of this study too. Again, probably similar results, probably will not change our practice. So I'll move to the novel agents now, and I think those are the new beta-lactams that are under clinical development, and some of them have already been FDA approved. And I've tried to put it in a table here that will summarize their activity against ESBLs, against AMC producers, serine carbapentamases, as been discussed here before, mostly the KPC, OXA48-like drugs, metallobeta-lactamases, Pseudomonas, and Acinetobacter. So I'll start, and I think if you know this this slide, and I think it needed a little bit of here, it should have been also FDA approved, but apart from that, I think it's quite accurate. If you look at that, I think that summarizes most of what we need to know about the new agents, and I will speak more about mostly those who are FDA approved. So Ceftolozane-tazobactam has activity against ESBL, does not offer more than other cephalosporins against the AMC or the carbapentamases, and has excellent Pseudomonas activity. Ceftazidime-avibactam, the hallmark is ESBL, AMC, and serine carbapentamases activity. And aztreonam-avibactam is under development. A phase 3 study just started. And what it will offer is a better MBL activity. Imipenem relebactam and meropenem vabrobactam are actually a combination of carbapenem with a new beta-lactamase inhibitor, and they offer excellent ESBL AMC activity and serine carbapentamases activity. And then I would go to Cefiderocol, the Shinogi compound, which I think is very interesting, although we know still quite not much about it. But what it offers primarily is across-the-board activity against all mechanisms of resistance, and very importantly, also cover Acinetobacter that none of the other beta-lactamase inhibitor is offering. So I will start with ceftolozane. Ceftolozane was developed as CXA101. It's a novel broad-spectrum cephalosporin with potent anti-pseudomonal activity. So the hallmark is actually that it's the best cephalosporin or the best beta-lactam against Pseudomonas. And what you can see here is susceptibility of different isolates, so all isolates, and that's ceftolozane, that's ceftazidime, and cefepime. And you can see percent susceptibility, and then when you go, and MIC 90, which is very low to ceftolozane, and when we go to specific mechanisms of resistance, so hyperproducers of AMC still maintain very high susceptibilities. Hyperproducers of efflux pumps, again, very high activity. So really, it's not affected by the common mechanisms of resistance in Pseudomonas to beta-lactam agents. So the problem is that ceftolozane is hydrolyzed by ESBL like other cephalosporins, so it was combined with tazobactam to block those ESBLs, and it provides a fair blockage of the ESBLs and protects ceftolozane. So here in this study, again showing the very good activity of ceftolozane against AMC hyperproducers, and with different mutation, and still the activity is maintained very nicely of the ceftolozane. And this study is very interesting because what it looks at effect of mutations to increase the MIC of ceftolozane, and what you see here is serial transfers in increasing concentrations of the drug, and you see ciprofloxacin, ceftazidime-avibactam, and ceftolozane-tazobactam, and you can see the rapid emergence of resistance within few days for these three agents, but very slow emergence of resistance, and truly not real emergence of resistance to the MIC of 4, even after 7 days with ceftolozane-tazobactam. I want to mention here that that does not mean that ceftolozane-tazobactam will not emerge ever, because if you take hypermutator strains, that will translate into multiple mutations and then a resistance, but that will happen only in a subset of the strains and only under certain conditions, and it's much more difficult to get resistance. So again, here it's another study that looks at the activity of ceftolozane-tazobactam against resistant Pseudomonas strains, and what they looked here is multidrug resistant, extremely drug resistant, and ceftazidime non-susceptible and susceptible. And what you can see here, that's the cumulative inhibition at different MICs, and what you can see here that in ceftazidime, for example, non-susceptible strains, at MIC of 4, which is the breakpoint for ceftolozane-tazobactam for Pseudomonas, you have 77% of the ceftazidime non-susceptible strain inhibited by ceftolozane-tazobactam. And again, for meropenem non-susceptible, again, very high rate, 85% of them are inhibited. So it offers excellent activity against non-susceptible Pseudomonas strains. And it has been licensed for intra-abdominal infections and for complicated UTI, and there is an ongoing trial for ventilator-associated pneumonia, just to mention that the dosage is different, that there is a double dose for ventilator-associated pneumonia than for the other indications. Ceftazidime-avibactam is also a licensed new medication. It's a strong, the avibactam is a strong inhibitor of class A and class C enzymes, and you can see here the inhibitor concentration 50, and the lowest is better. And what you see here is compared to clavulanic acid, tazobactam, and sulbactam, much lower IC50s, and especially it's important to see for the KPC a very nice inhibition by avibactam. And the same for the class C, it's a very good inhibitor of class C enzymes. So when we look at the ceftazidime-avibactam spectrum of activity, it will cover the class A, that's what tazobactam will cover, or clavulanic acid, and it adds the KPC enzyme, it adds the AMC enzymes, and the class C enzymes, and part of the OXA, the part that is important in enterobacteriaceae, so the OXA48, OXA181, it does not have an effect on the OXAs in the Acinetobacter part. And it does not have an effect very importantly, on metallo-beta-lactamases. So just to show you the activity against KPC-producing strains, and that's ceftazidime-MIC, and you can see how the KPC destroys ceftazidime quite easily, and avibactam reconstitute the activity of ceftazidime. It was developed and approved for complicated UTI, intra-abdominal infection, and HAP-VAP, and had also a resistant pathogen study, but the resistant pathogen study aimed at ceftazidime-resistant strains, and obviously almost all of them were ESBLs or Pseudomonas that were resistant to ceftazidime, and very few CRE patients. Just to show you the results of the resistant pathogen study, here what you have is very similar clinical efficacy, and when you look at microbiological response, you see a better response of ceftazidime-avibactam than best available therapy, and the best available therapy was primarily carbapenems. What do we know about the CRE and ceftazidime-avibactam in real world? So there are several large-scale, I would say, reports on clinical experience, and overall there is a good success rate relative to the severity of the infection, about 70% response rate. And there are several cases of emergence of ceftazidime-avibactam resistance during or after therapy. Those were de novo mutation and the KPC enzymes that resulted in reduction in carbapenem-MIC, and apparently they did not translate into clinical failure, but I think the experience is still very limited, so we don't know enough about it. A very recent study on comparing Colistin to ceftazidime-avibactam treatment of infection due to CREs, and here what they did is they looked at 38 patients treated with ceftazidime-avibactam versus 99 patients treated with Colistin, and they looked at multiple outcomes, and those are actually mortality, and you see mortality has been reduced, and that's the good outcome is discharge home, and more patients were discharged home. And what you see that also patients who were discharged to other facilities, so long-term care facilities and others, will also improve. So overall, good response, but we have to remember we speak about very severe patients, so the fact that there is an improvement is nice. We could not expect 100% response rate with any of those drugs. So imipenem relebactam is developed as relebactam is a new beta-lactamase inhibitor, very similar in its activity to avibactam that I've described before, and it has completed phase 2 urinary tract infections and intra-abdominal infection, and it's undergoing phase 3 studies and a pathogen-directed study. It has good penetration to the lung, so we should expect that it will be active in VAP. And it has, you can see here, the effect of the addition of relebactam to imipenem, and you could see the MICs for the KPCs are dramatically moving to the susceptibility range. For MC, there is also an improvement, MC with efflux-PAM mutation, so CRE that are not carbapenemase producers. You can see also improvement because of the blockage of the MC. With the OXA48, you see that about half of the population, it has an effect, but half it does not have an effect. And that's probably because of the inhibition of the ESBLs. Most of the OXA48 carry also ESBL, so probably it's the effect on the ESBL. And then no effect in the metallo-beta-lactamases, so also this agent is not effective against metallo-beta-lactamases. For Pseudomonas, it will also offer an improved activity over that of imipenem, with about 50% of the imipenem-resistant strains, Pseudomonas strains, will become susceptible to imipenem-avibactam, and that's because two mechanisms are required in most cases, an OPRD loss and an AMP-C, and that blocks the AMP-C. Vabromer is a meropenem-avibactam combination. Again, a new inhibitor with very similar spectrum to the other that I've just described. And you can see a very nice effect on reduction of the MIC or recovery of the meropenem activity against KPC producers. And it's completed a phase 3 study, complicated urinary tract infection, compared to Ceftolozane-tazobactam, and actually showed superiority. It's also completed a pathogen-directed CRE study compared to best available therapy and showed superiority to the best available therapy. So it was licensed by the FDA, but it's moving from one company to another, and we don't know how it will be developed or if it will be developed and will reach clinical use. Cefiderocol, that's the Shinogi compound that I mentioned before. It's a siderophore cephalosporin. It uses iron transport mechanisms to go into the cell, and called by that Trojan horse. It's highly stable to all beta-lactamases, and it has activity against multidrug-resistant Acinetobacter and Pseudomonas. And you can see here the activity against different, in large number of different strains, Enterobacteriaceae, Acinetobacter, and Pseudomonas, and you can see very low MICs to all of those. And I think the main important addition will be the activity against Acinetobacter and maybe metallobeta-lactamases also. It completed successfully a phase 2 complicated urinary tract infection study, and there are ongoing studies, phase 3 studies, pathogen-directed study, and a HAP-VAP study, so we still have to wait and see. There were issues with some side effects, and we don't know whether it will succeed in development or not. There are also several non-beta-lactam agents that are under development. There is Eravacycline, which is a tetracycline derivative with very nice activity across the board, except it does not have any activity against Pseudomonas, but importantly, it has effect against Acinetobacter. And Plazomicin, which is an improved aminoglycoside, again with very nice activity, but it's an aminoglycoside. And Murepavadin, which is a new drug directed just at Pseudomonas, and I'll start with that. It's a first in the class cyclopeptide, specifically targeting the membrane of Pseudomonas. It reaches a high level in the lungs, but we are still at early stage. We don't know if it will be developed, in what way, and how will we use it. It will be only Pseudomonas-directed therapy. Eravacycline, as I said, is a very interesting in terms of a broad spectrum activity, and especially its activity against metallobeta-lactamases and Acinetobacter. I think that it's important to remember that it's a tetracycline derivative, so we should I think that we should take the knowledge about what we know about tetracycline and expect that similarly it will have similar uses at the end. Indeed, the company conducted 2 studies on on urinary tract infections, and both failed, reaching the primary endpoint, which most of us would have guessed that will happen with the tetracycline. But I still think it will be a very important drug, definitely for skin soft tissue infections, and hopefully also for lung infections. And plazomicin is a semi-synthetic aminoglycoside. It's stable against all transferable aminoglycoside modifying enzymes, but not against ribosomal methyltransferases. Unfortunately, many of the NDM strains have been reported to have ribosomal methyltransferases on the same plasmid, so that will be a limitation. Overall, it had completed successfully phase 2 studies and completed a phase 3 CRE study with actually showing superiority against best available therapy, which was mostly Colistin or Colistin combinations. And you can see here the 28 all-cause mortality in the plasmomycin arm was 28% versus 50% in the best available therapy, and that's truly the mortality, and that's also complications. Apparently, many of the studies that have been done until today on the CRE are stained with Colistin-resistant strain being reported as susceptible and included in the studies. So many patients were actually probably treated with placebo. So we have to take it into account in interpreting the results for the future. So I'd like to summarize here. There are new treatment modalities that are emerging. As was said here before, Colistin will likely disappear. I don't know if that's true. I've looked at Mark's slide on use of anti-MRSA drugs. Most of us would have said vancomycin will disappear from the world, but still, most of our patients with MRSA are treated with vancomycin. And but truly, I think that Colistin should disappear. We have to have alternatives, but it should disappear. It's a non-efficacious drug. So new beta-lactamase inhibitors are arriving and expand the treatment options against multidrug resistant gram-negatives. New cephalosporins and new non-beta-lactam agents are also coming. I think that we will have good treatment now, or have good treatment now, for multidrug resistant Pseudomonas, for KPC producing strains. We don't have yet very good options for metallobeta-lactamase producing organisms and for multidrug resistant Acinetobacter. So thank you very much.